Now these mediators are released either from the cells or are derived from plasma proteins. Cell-derived mediators are released either from their storage in the cell granules or are synthesized in the cells, or the most common site of synthesis of plasma-derived mediators is the liver. After their release from the liver, these mediators require activation. So these are the few general features of mediators. Now these are the different responses on which all mediators are released. These basically are the general stimuli. These stimuli may be a variety of injurious agents. It could be dead bacteria, it could be damaged tissues, it could be even one mediator stimulating other mediator, and the later may be called secondary mediator, which may perform the function of initial mediator or may have the opposing action, right? And this is a diagram, table in which you can have a quick review over the source mediators and main action of different chemical mediators, right? So basically it is divided into cell-derived and the plasma protein derived. It means some cells are secreting chemical mediators like mast cells, basophil, platelets, and inflammatory cells like lymphocytes, leukocytes, and monocytes. So mediators are the histamine, serotonin, prostaglandins, leukotrienes, and the main function is mostly increase in the permeability and vasodilation. While plasma-derived proteins are source is the plasma, and they have clotting and fibrinolytic system, Kinin system, complement system, which is further activating few other chemical mediators, and in return basically they act on the vessels and causes permeability. Now the target organs of chemical mediators are different, and they may have similar action or they may have different action in different target organ cells. They may act on cells which formed them or maybe on the other body cells. Now what could be the range of actions? What are the different actions of chemical mediators? Number one is increased vascular permeability. It has vasodilatation effect. It has chemotactic effect. Some mediators cause fever and pain, and some mediators are released for the tissue damage, to damage the bactericidal or the injurious agent. Apart from injurious agent, it has to destroy the tissues on which the injurious agent is acting. Mediators have short lifespan. After their release, they are rapidly removed from the body by various mechanisms. For example, by enzymatic inactivation, antioxidants sometimes, and sometimes they are regulatory proteins or may even decay spontaneously. So these are the different things that remove chemical mediators, and that's why it won't causes any other further effects. Now, as we have discussed that chemical mediators are divided into major two groups. One is cell-derived mediator and the other one is plasma mediator. In cell-derived mediator, we have further divided into vasoactive amines. In vasoactive amines, we have three different mediators. These in which we have two important pharmacologically active amines that have a role in the early inflammatory response, basically in the first 1 hour, is histamine and serotonin, that is 5-hydroxytryptamine. Then third one is the neuropeptides. It has the similar action, but it has a in the lower intensity as compared to the histamine and serotonin. Now, first of all, histamine. To remember any chemical mediators, you should remember on three basic features of any chemical mediator. One is from which it is released. What is the number two is the stimuli, and number three could be the action or what could be the effect. So histamine is released from mast cells, basophils, and platelets, and stimulus on which histamine released is mostly acute inflammation, in which we have heat and cold or irradiations due to any trauma, due to any irritant chemicals, or it could be related with immunological reaction. These are all the causes of acute inflammation. Sometimes it also secretes by some anaphylatoxins, which itself is chemical mediators, like C3A and C5A. These are the complement system, which you will study in the next slide. The anaphylatoxins also releases anaphylatoxin. These two, C3A and C5A, counts as anaphylatoxins, and these anaphylatoxins will secrete histamine, and which in return increases the vascular permeability and causes edema in tissues. So these are the stimulus. Some other stimulus in which histamine releasing factors from neutrophils, monocytes, and platelets are also there. Then we have interleukins. So this is basically the stimuli. Now the main action of histamine is to dilate vessels, that is vasodilation, and increase vascular permeability. Apart from these two, itching and pain is also sometimes caused by histamine. Now the second entity is serotonin or 5-hydroxytryptamine, which is released from the chromaffin cells of GIT, spleen, nervous system, mast cells, and platelets. The action of serotonin is similar to histamine, but it is a less potent mediator of increased vascular permeability and vasodilation than histamine. And one thing I should mention it here, that the carcinoid tumor, which is the most common one in appendix, and it is a tumor which releases serotonin. So carcinoid tumor is a serotonin secreting tumor. Now the third entity for chemical mediators, which are vasoactive or vasoactive amines, is neuropeptide. Neuropeptides are the small peptides which are released from central and peripheral nervous system, and the major or most common action of neuropeptide is increased vascular permeability, transmission of pain stimuli, and mast cell degranulation. Now the second group of chemical mediators, apart from vasoactive amines, is arachidonic acid metabolite. The second group, the first one was vasoactive amine, the second one is arachidonic acid metabolites. Basically, arachidonic acid metabolites are the most potent mediators of inflammation, and much more than the oxygen-free radicals. These are the major potent chemical mediators as compared to oxygen-free radicals. Oxygen-free radicals basically are the radicals which forms due to any continuous oxygen and anaerobic cycles. As you must have learned about the causes of cell injury is oxygen-free radical. So arachidonic acids are those metabolites which are more potent as compared to oxygen-free radicals. It means it causes much more inflammatory effect or causes damage to the tissues. Arachidonic acid is released from the cell membrane by phospholipases, and it is then activated to form arachidonic acid metabolites. Arachidonic acid metabolites are formed via two pathways. One is cyclooxygenase pathway and other one is the lipoxygenase pathway. Each pathway gives many metabolites, many chemical mediators which causes different effects. This is the production of arachidonic acid metabolites and their role in inflammation. You can see cell membrane, and in cell membrane there is phospholipids. Phospholipids via phospholipases forms arachidonic acid, and then further arachidonic acid is producing through cyclooxygenase pathway and lipoxygenase pathway different chemical mediators. Through cyclooxygenase pathway it forms prostaglandins and thromboxane A2, while through lipoxygenase pathway it forms leukotrienes, and in which we have leukotriene A4, B4, C4, T4, A4. So basically, instead of remembering all the chemical mediators as such and then remember the action, it is very difficult. My advice to all people, first remember or first recall the actions. What are the different actions that you need to cover in inflammation, like cellular events and in vascular events. So there are two main events, and which further what are the different events happen in inflammation, and what are the main chemical mediators which help in specific actions. So then you can easily remember what are the chemical mediators and how you can easily remember, right? Now the first pathway is cyclooxygenase pathway. In cyclooxygenase pathway, the activated arachidonic acid, which is secreted from the cell membrane due to acute inflammatory action, it forms PGG2, that is prostaglandin G2, then prostaglandin H2 it forms, and then combines with free radicals. It gives many other metabolites like prostaglandin G2, that is PGT2, PGE2, which causes vasodilators, which are the vasodilators, bronchodilators, and increase permeability. The further are PGF2 alpha, thromboxane TXA2, PGI2 resolvents. These are the different metabolites. Just remember the action of these metabolites, in which thromboxane has approximately opposite effect of other prostaglandins, like vasoconstriction. Apart from vasoconstriction, it also causes bronchoconstriction and platelet aggregation, while others have the opposite effect, like PGD2, PGE2 is bronchodilators and vasodilators and bronchodilators, while PGI2 is bronchodilator, vasodilator, and anti-aggregating agent. So anti-aggregating agent means it is against the platelet aggregation. So just remember that thromboxane A2 causes vasoconstriction, bronchoconstriction, and platelet aggregation. It enhances the plaque formation, right? So these are the cyclooxygenase pathway and its metabolite. Now the second pathway for arachidonic acid metabolite formation is lipoxygenase pathway. In this pathway, lipoxygenase enzyme is utilized after activation of or the secretion of arachidonic acid from the cell membrane. It turns into HPETE which is a potent chemotactic factor. Then it gives LTA4, that is leukotriene A4, then LTB4 and LTC4, LTD4, LTE4 and lipoxins. So LTB4 is a potent chemotactic factor, as well as it causes cell adherence of different cells to the cell wall, as you must have learned about the margination, rolling and chemotaxis in the process of acute inflammation. Then LTC4, LTD4, LTE4 and lipoxin causes smooth muscle constriction, vasoconstriction, bronchoconstriction and increased vascular permeability. So if you remember all the sequences of processes which happens in acute inflammation, you can easily figure it out, or you can easily remember the affiliated chemical mediators with the process, inshallah. Now the lysosomal component. Lysosomal components basically are the lysosomal granules, which are secreted from the neutrophil and monocytes. And these lysosomal granules are released and elaborate a variety of mediators of inflammation. Now, the neutrophil or the lysosomal components which are secreted by neutrophils are majorly myeloperoxidases. These myeloperoxidases are stored in the granules of neutrophils, in primary, secondary and tertiary granules. And as you remember, the granules of neutrophils are azerophilic. Both are pink and purple coloration, right? So these granules contain myeloperoxidases. When this myeloperoxidases releases, it causes different actions like degradation of extracellular components like collagen, basement membrane, fibrin and cartilage, as well as, of course, it kills or destructs the bacteria in phagolysosomes. But the degradation of extracellular components like collagen, basement membrane and fibrin should be stopped here. Otherwise, there will be a harmful tissue destruction in body will occur. So for this purpose, or for the anti-myeloperoxidases, there is some anti-proteases called alpha-1 antitrypsin and alpha-2 macroglobulin. So these are the anti-proteases which stops the function of myeloperoxidases which release from neutrophils. Then comes the granules of monocytes and tissue macrophages. Basically, these granules are majorly secreted during the chronic inflammation as compared to acute one. Majorly it is secreted in chronic inflammation. And the function, of course, it is related with the cells on degranulation, tissue and killing of the bacteria, and it is also destructing the surrounding tissue. So these are the granules of monocytes. Now the fourth chemical mediator is platelet activating factor, or PAF. It is released from IgE sensitized basophils or mast cells, or it could be released by other leukocytes, endothelium and platelets as well. Apart from its action on platelet aggregation and release reaction, the action of PAF as a mediator of inflammation also has these following actions, like increased vascular permeability, It also causes vasodilation in low concentration and vasoconstriction. It causes bronchoconstriction. It causes adhesion of leukocytes to endothelium or aggregation of leukocytes and chemotaxis. So these are the major actions of platelet activating factor. Now the fifth cell-derived chemical mediator is cytokines and chemokines. Cytokines are polypeptide substances produced mainly by activated lymphocytes and activated monocytes. So those which are secreted from lymphocytes are called lymphokines and monocytes from monokines. While the term chemokine is used for a family of substances which acts as chemoattractant for inflammatory cells. Now these are the main major cytokines in inflammation, in which we have interleukin 1, which is secreted from monocytes, macrophages, B cells, lymphocytes, and the target cells are all cells, and the main function is in fever and shock. It gives expression of adhesion molecules, and it immigrates neutrophils and macrophages towards interstitial space, if you remember immigration process. Then interleukin 6 is same as source is same as interleukin 1, but the cell target is T and B cells, and basically main function is acute phase protein production, which is C-reactive protein, and differentiation and growth of T and B cells. Interleukin 8 is monocyte macrophages secretion, and then cell target is neutrophils, basophils, T cells, monocytes, and it induces migration of the neutrophils, macrophages, and T cells. Right? Then interleukin 12, the main action is to help formation of T helper cells and killer cells, and increases production of interferon gamma. Interferon gamma is necessary for viral infections. We usually do it for viral infection. And just remember why we were studying acute inflammation and chronic inflammation. You will study in chronic inflammation that what are the major predominant cells of inflammation. Of course, for acute inflammation there is neutrophil, macrophages involvement, while for the chronic inflammation you will have the lymphocytes, fibroblast, plasma cells. So whenever you will study chemical mediators, then remember what are the major chemical mediators which helps in acute inflammation and what are the different chemical mediators which helps in chronic inflammation. So chronic inflammation, we have interleukin 12 because it acts on T cell formation. T cells are the lymphocytes, and it increases in viral infections and chronic infections. While in interleukin 17, CD4 T cells, it is secreted from T cells, and it increases secretions of other cytokines and migration of neutrophils. So in this chart, basically you have most important cytokines which you should remember is interleukin 1, TNF gamma, interferon IFN gamma, that is TNF alpha and interferon gamma. So tumor necrosis factor alpha, interleukin 1, INF alpha, interleukin 6 and 8. These are also comes into the important categories. So this is about the major cytokines in inflammation. Now this table shows you what are the main cytokines which are present in acute inflammation and those which are present in chronic inflammation. For acute inflammation we have TNF, interleukin 1, interleukin 6, chemokines and interleukin 17. While in chronic inflammation we have IFN gamma, that is interferon gamma, interleukin 17, interleukin 12, right? So just remember the cytokines which are involved in acute inflammation mostly and in chronic inflammation, and remember the actions in inflammation, right? Now this diagram shows you the major functions of cytokines in acute inflammation. They have taken three chemokines like TNF and interleukin 1. What are the local inflammation effects of TNF and interleukin 1? It increases expression of adhesion molecules and it increases permeability, right? So it causes edema and immigration and rolling process, which you have learned in the vascular events, right? Then leukocytes, TNF also acts on leukocytes to further secrete interleukin 1 and interleukin 6, and this is the local inflammation effects of TNF and interleukin 1. So majorly TNF and interleukin 1 are the chemokines which majorly acts on acute inflammation. Then comes the systemic. What are the different systemic protective effects of TNF and interleukin 1? It acts on brain and it causes fever, so increase in the temperature causes to kill the bacteria and microorganisms if they are. Then liver is from liver, it secretes different acute phase proteins which are C-reactive proteins, right? And which helps in degradation of the microorganisms and forming the antibodies and different. For every purpose we need proteins, right? So it acute or it increases acute phase proteins. Then on bone marrow it acts as a leukocyte productive mechanism. It increases leukocyte production. So this is the systemic protective effect. While the systemic pathological effect, it acts on heart, TNF, and increases it decreases basically low output. It decreases cardiac output by vasoconstrictions, right? Then on endothelial cells, it causes or it increases thrombus formation and increase permeability. So basically, it decreases output of blood vessels and apart from vasoconstriction. Then there comes the multiple tissue on different tissues like skeletal muscle. It acts as an insulin resistant chemokine, so glucose can be utilized where the energy is needed during the acute inflammation instead of giving the skeletal muscle, which is not needed right now, right? So it for major organs like liver, heart, brain. So it preserves energy by increasing the resistance of insulin to the skeletal muscles. Now the second group of chemical mediator which are derived directly from plasma proteins. These basically are the proteins which includes different products derived from activation and interaction of 4 interlinked systems, in which we have kinin, clotting, fibrinolytic, and complement system. Each of these systems has its inhibitors and accelerators in plasma. With negative and positive feedback mechanisms. In this chemical mediator, the Higman factor, which is factor 12 of the clotting system, plays a key role in interactions of the 4 systems. These activation of factor 12 in vivo by contact with basement membrane and bacterial endotoxins, and in vitro with glass or kaolin leads to activation of clotting, fibrinolytic and kinin systems, right? So these are the two activating reasons in vivo. In vivo means when it is present in body, and in vitro means when it is outside the body, right? So in inflammation, activation of factor 12 is brought about by contact of the factors leaking through endothelial gaps. The end product of this activated clotting, fibrinolytic and kinin systems activate the complement system that generate permeability factors, and these permeability factors in turn further activate clotting system, right? So this is the general features of plasma derived protein mediators. Now this is the interrelationship among clotting, fibrinolytic, kinin and complement system. It starts from the activation of factor 12. Then activated form is called factor 12A when it contacts with the in vitro in vivo stimulating factors. Then after activation of factor 12, it can go to clotting system, it can produce kinin system and it can produce fibrinolytic system. Then first fibrinolytic system could be turn up into plasmin. This plasmin activates fibrin and clotting complement system, and in return it causes permeability factors, that is C3A, C5A. These are the complement factors. Then the clotting system, in clotting system of course it starts from fibrin production, then fibrin split products occur, then complement system activation occurs, and then permeability factors happens. Then kinin system forms calcarein and bradykinin. This calcarein directly acts on permeability factors instead of activation of complement system. So this is the interrelationship among clotting, fibrinolytic, kinin and complement systems, right? Now the first chemical mediator is the kinin system of plasma derived chemical mediators. Now this system on activation by factor 12A generates bradykinin, and after as the name implies that bradykinin means, brady means slow and kinin means active, so it basically slows the contraction of smooth muscles. First calcarein is formed from plasma precalcarein by the action of precalcarein activator, and this bradykinin basically it acts in the early stage of inflammation, and its effects include smooth muscle contraction, vasodilation, increased vascular permeability and pain. So these are the effects of kinin system. Now the second entity is clotting system, and in clotting system the pathway occurs it starts with the activation of factor 12A. It initiates the cascade of clotting system, the whole clotting system with 9, 11 clotting factor activation and so on and so forth, resulting in the formation of fibrinogen, which is the clotting factor 1, which is acted upon by the thrombin to form fibrin, and the action of fibrinopeptides in inflammation are increased. Basically clotting factor, what is the function of clotting system? It increased vascular permeability, it chemotaxis for leukocytes, and it is an anticoagulant. It has also an anticoagulant activity. So this is the pathway of clotting system. So now on this note, you can see that there are both factors, or both the things is happening in this clotting system. Apart from clotting, there is an anticoagulant activity. It means when the prothrombin and thrombin forms, this occludes or closes the vessel lumen by the formation of fibrin from fibrinogen. Once fibrinogen forms fibrin and there is an increased amount of fibrin, Then the fibrinogen activates plasmin, and this plasmin is called fibrinolytic system, which activates fibrin peptide. So it clears the fibrin and further decreases the fibrin formation. Now the third system is activation of fibrinolytic system. What is the purpose of fibrinolytic system? Basically, it activate factor 12 to form further precalculin system to generate bradykinin. Number 2, it splits off complement C3 to form C3A, which is permeability factor, and it degrades fibrin to form fibrin split products, which increases vascular permeability and chemotactic to leukocytes. So these are the things that caused by the fibrinolytic system. Basically, fibrinolytic system activates these systems to further clarify the pathway or vessels, and it is basically a fibrinolytic system in which fibrin deposition decreases. Now the last system is the complement system. This complement system activates by two methods or two pathways. One is the classical pathway, through which antigen-antibody complexes are formed, and then it activates this system, complement system. The other system is the other pathway is called alternate pathway. This activates via non-immunological agents such as bacterial toxin, cobra venom, and IgA. So it doesn't need any immunological reaction like antigen-antibody complexes. It activates directly by venoms, by toxins, or any other secretory IgA bodies which are formed in autoimmune diseases. So complement systems on activation by either of these two pathways, what yields? It yields activate products which includes anaphylotoxins, that is C3A, C5A, and C4A. And these are basically all called anaphylotoxins. And apart from this, it also forms membrane attached complexes, that is MAC, which includes C5b, C6, 7, 8, and 9. Now, what are the different actions of these activated complement systems in inflammation? It is in C3A, C5A, and C4A, it basically anaphylotoxins activates mast cells and basophils to release histamine and causes increased vascular permeability and causing edema in tissues. It also augments phagocytosis. C3b is an opsonin which covers the antigen to make it delicious for opsonization, right? For phagocytosis, sorry. So this is called opsonization. Then C5a is a chemotactic for leukocytes, while the membrane attack complexes, which is C5b to C7, is a lipid dissolving agent and causes holes in the phospholipid membrane of the cells. Or of the cells of bacteria, or it could be related with our body cells. So these are the complement system and how they activate. Now this is the role of mediators in different reactions of inflammation. You can see chemical mediators in the other way around. First you can see the reaction of inflammations. What are the different actions and what are the principal mediators. In vasodilatation you have histamine and prostaglandin for increased permeability. This is C3A, C5A, and histamine and serotonin, and so on. Then chemotaxis, leukocyte recruitment for TNF and interleukin-1 and activation. Then for fever it is interleukin-1 and TNF, and for pain this is mainly prostaglandins and bradykinin. While for tissue damage, this is lysosomal enzymes of leukocytes and reactive oxygen species. So this was all about the chemical mediators. I hope I can help you in studies. This lecture will help you. And this is not all the chemical mediators that you can study. for increasing your horizon, for increasing your knowledge. You have to study from your books, and I'm just giving you a short brief, rather trying my best to give you a brief summary of chemical mediators, but there are lots and lots of things you need to study by yourself. Thank you so much.